Yeah, so welcome everybody. I hope you can see my screen here. What I wanted to do today was really just kind of go over a bunch of different research projects that we've got going on here with Extension that deal with invasive species. So I'm just going to kind of hit each one short, short and quick, but I think they're useful. It kind of highlights what we do with Extension, how we use research and tie it in with our outreach programs, and then really how we try to make our research very applied, very useful for the people of Illinois. So why do we do research? Really the bedrock of Extension, if you're unfamiliar with it as a program, is the transfer of unbiased, evidence-based information to landowners, land managers, and producers. We take the science from the scientists and give it to the people and show them kind of how to use it, what it's there for. Again, it's really transferring that information to the best we can and giving the best recommendations and the best information to the people of Illinois. And our information we give really needs to be backed by sound research and sound science. And so oftentimes we use other people's research, whether they're at the university or other universities, but Extension does a lot of research ourselves as well to answer those questions that we need answers for. And this happens in a lot of different arenas, including natural resources. So the research in natural resources, we're really trying to better understand the natural world. And that could be to help protection of it, stewardship of it, management of it, restoration of it, different aspects. We're trying to answer these questions that'll help us understand more about the natural world and figure out how to manage them. Here's just a shot I threw in there of Nick doing some of his chaff flower research. So he's a member of the research group here in Southern Illinois. But our research, goes to presentations, talks, outreach, as well as publications like the ones you see up here now, where we're tracking invasive species, forest health issues, everything out there. And so today I'm going to focus on our invasive plant research and some of the stuff that we have going on. And the first is a pilot project that really Nick and then a fellow that works with me, Kevin Rowling, are taking the lead on, but I'm somewhat involved, so we wanted to add it here, is this UAV or drone research that we've got going on, trying to see how we can use those to better detect and map invasive plants in forested ecosystems. It's a neat idea. The idea is making it more efficient, and we can get out there and access sites that we otherwise can't easily access. And so we started this pilot project just to try to understand what's the inputs with using a drone, what are the results, are the results accurate enough, or kind of how best can you use this tool for invasive species. And so in terms of equipment, we have a Phantom 4 Pro version 2 drone. We use Drone Deploy, which is the program that lets us create our photo mosaics, as well as ArcGIS and Collector. We use a couple other of those mapping programs here. And so what we do, instead of just flying up and taking pictures with the drone or flying up and looking through the camera of the drone, we are instead actually having the drone program in a flight for the drone. So it flies itself through a grid where it's taking pictures throughout the whole grid, and then builds this huge mosaic of imagery that can be geo-referenced and put in there so you know where it's at in the world. And it has this stitched image from hundreds of images together, where you have this high resolution map based on all these images. And again, we basically program it in, we set it on the ground and hit start, and it flies and it makes and does this itself, which is a nice thing. So it builds this grid. We have to set the boundaries there. We have to set how much overlap in the images, what altitude, what's the different terrain that we're dealing with, the direction of wind. All of these things are into account into making the best imagery. And then at the end, we have this single image layer that we go on there. And so this is kind of what we do. And then you can either do auto recognition through looking through certain color spectrum, but then what we often do is just visually searching this imagery to try to find the clues for the invasive species. So we wanted to see if this was useful and how efficient this was for looking at sites and trying to pick out invasive species. And so we tested this by looking at 13 5-acre blocks here at the Ag Center where we captured imagery twice. We tried to time it for leaf off when the overstory trees lost their leaves, but particularly we're looking at Amur honeysuckle when it was still green and identifiable. And so we did this in the spring and in the fall, and we both captured the imagery with the drone and then mapped what we could find there, as well as capturing the imagery or capturing the mapping of these invasives through walking grid surveys. And so we looked at both of those and how efficient they were and how accurate they were and compared them. In terms of efficiency, walking all of these in grids and mapping them was much less efficient. So it took to do all of these 13 sites about almost 12 hours, whereas the drone was able to do it in under 2 hours. So it was very, very efficient. We did have a little extra time processing the imagery and things, but it really was kind of did that mostly on its own. And so we found out it was very quick. In a matter of less than 10 minutes, you can do a 5-acre block and get that imagery in the field, which was very, very nice. In terms of accuracy, we found that timing was very critical with this, where if you missed it a little bit on the front end and you had too many leaves on the trees, you had a hard time seeing the honeysuckle. But if you waited a little too late and too many of the leaves were falling off or they started really yellowing, they were hard to pick up. So while it was very useful, there was a tight window in the fall, and in the spring that window was even tighter or maybe non-existent. So while we did have some issues finding the timing, if you could hit it right, the honeysuckle was identifiable. In terms of what the accuracy of this and what was identified, we found that we did misidentify some larger Japanese honeysuckle stands as bush honeysuckle. They looked a lot from the same from the air. And we also had trouble picking up smaller or top-killed individuals or young or small bush honeysuckle plants. They just didn't show up on the imagery. But where we did have good luck in finding them was larger single bushes or small stands of or small infestations of honeysuckle. We were able to pick it out. We're still going to keep looking at this, and we're still going to try to refine our timing and refine our processes to do this. But overall, what we found with this, again, is timing is critical, but it does have a lot of application for identifying and delineating these large stands of bush honeysuckle or calculating acreages for sure to try to figure out kind of the scope or the scale of the problem you have of honeysuckle. So while it may not be useful for early detection or identifying these small stands, it does have some usefulness, and we're going to continue to try to figure this out. We also want to look at this for other species. We think there's a lot of use for this for other species that are maybe even more identifiable than honeysuckle. Here we have a lot of princess tree or paulownia, and in the spring it blooms with those bright purple, kind of violet purple flowers at the top of the trees, readily identifiable at that time. So we're going to try to fly this spring when they are flowering to see how useful it is for identifying them. And then also some of the vine invaders. So this picture here was actually taken from an airplane, and it is all Oriental bittersweet, and this is in the Northeast, but readily identifiable in the larger infestations from the air. So we're going to start looking at maybe that, look at some other species as well, just to try to find out where this technology can be best applied. So that's just, again, a pilot project. We're just getting way under that. Excited about it. I'm looking forward to kind of how that develops, and particularly Kevin and Nick, how they continue to develop that research moving forward. What I'm going to spend the rest of the time on is some of the control research that we do here, and our goal with this control research is really to refine these management recommendations. Trying to get effective, safe methods to control these invasive species. And so we're looking at herbicides and rates and timings. Again, trying to refine and find out what works the best for the species that we deal with. And just a little plug, some of the information that we're doing and we're collecting with this research, as well as others, has went into our publication, The Management of Invasive Plants and Pests of Illinois. So we do have plenty of copies of those. So if you're interested in this booklet, I think it covers 42 different invasive plants here in Illinois. Get a hold of me and we can get you some of these books. It's also available on our Extension Forestry website. But one of the neat things we added in this book through a lot of observational kind of citizen scientist projects is this phenology calendar. And this lets us get a better idea of when the timing of the development throughout the year of the different invasive plants and how it differs across Illinois. So again, it helps us refine when to look for them, when to control them. But in terms of our research, again, we're looking at methods, timing, rates, and herbicides for these different invasive plants. The three projects I'm going to talk about today, one is a garlic mustard timing research, looking at when you can control them and what's the best time. Look at pre-emergent treatments for Japanese stiltgrass, and then bush honeysuckle fall foliar treatments, and then we're just starting a burning bush kind of similar project for the honeysuckle, which I won't talk about today. But just jumping in, garlic mustard, most of us, since we're plant people, know garlic mustard and know that it's an issue throughout the state, particularly here in southern Illinois. We have problems with it early in the spring, where it could impact some of our native spring wildflowers. It is considered a high priority species for the state. And again, you know, if you're not familiar with it, you can find it, unfortunately, fairly widespread throughout the area. But one of the issues with garlic mustard is the thought has been there's been a tight window in the spring where you can use herbicides. And so if you look at some of the conventional control recommendations that are out there in publications, they say things like the application should be made to the foliage in the spring when the plants are in the rosette stage, so when they're not flowering, or application should be made early spring prior to flowering. So basically you've got a tight window from when things green up, the plant gets going, until it flowers, to spray these. And after that, oftentimes what is recommended and what we move to is hand control, pulling them up, bagging them, pulling them out. And that's very effective, but it's also very labor intensive. It takes a while, and so it slows you down on the amount of garlic mustard that you can control in an area. So what we wanted to look at was, is that recommendation for stopping herbicides when the plants start flowering, is that a valid recommendation, or can you actually go a little longer than that? Kind of how effective is that? So specifically, we're looking at, can you make these treatments later than what is normally recommended? In other words, past the start of fruit development. So there's already fruit on the plants, and can those treatments be effective, and will the seeds that are already there be viable, or does the treatments kill those seeds? And what we're trying to do is expand that window in the spring when you can use herbicides. We did this in partnership with the University of Wisconsin, and we had small plots and 3 to 4 replications in 3 different sites. And we looked at glyphosate, which is Roundup, at 3%, triclopyr, which is Garlon, and a bunch of others at 1%, which both of those are kind of the common recommendations, as well as untreated control. Again, we did this 3 places, 2 in southern Wisconsin and 1 down here in southern Illinois. And we waited until the plants, the garlic mustard plants, had finished flowering, and really their last petals were falling off the plants before we treated those. And so that was May 7th down here in southern Illinois. And then in southern Wisconsin, that was May 27th and then May 31st. So you can see a little bit difference in timing. We did that because we were focused on the development of the plants, and we wanted to spray them at that point when they just stopped flowering. And so here's a shot of some of the garlic mustard down here. And you can see there's still some flowers on it at the main top of the plants, but there's still a lot of, already a lot of seeds on there. The saliques, the seed pods are numerous on the plants. They're large. So it's well past the start of flowering, and it's kind of at the end of flowering. We waited one more week past this to let the small flowers at the top go ahead and finish before we sprayed them. And what we found was the glyphosate, so the Roundup, the glyphosate really browned these plants up fast. So this was 2 weeks after treatment, and you can see everything's down and dead. Now there's still a lot of seed pods on those plants, right? The seed pods were already there, but the plants kind of died quickly in terms of defoliated quickly after the treatment. And it was a little different with the triclopyr. They 2 weeks after treatment, you can obviously see they've been sprayed, but it was a little slower acting, and it waited until about 6 weeks after treatment really to see them completely defoliated. But again, we didn't really care necessarily about how quick they were defoliated. Our concern with garlic mustard, since it's a biennial, was getting rid of those seeds. That was the most important part in terms of what determined the success of this, this research and this treatment. So what we did was cut and bag 3 plants from each plot. We dried the bags until the seed pods were fully dry, and then we collected the seed from those, weighed all the seed, and then estimated the total number of seeds based upon a subsample count. And then we tested the seeds for viability using a tetrazolium test, which is where you stain them. If they turn a certain color, it shows that they are respiring, and so that they're still a viable seed. And so we wanted to get from this basically the number of seeds as well as the number of viable seeds that each plot produced. So this is what we found. It was a bit variable, depending on the site. But overall, both treatments, even that late, were very, very effective at reducing the number of viable seeds at each of our treatment sites. And so one site, it was only 70% reduction with glyphosate. But other than that, they were all well over 80, and most of them were in the 90s. And especially here in Southern Illinois, ours, there was almost no viable seed, even spraying it at that time. So that was pretty promising. We definitely a reduction in seed, but not a complete elimination. And so kind of in summary from that, these late treatments of both herbicide types we used were very effective at reducing the amount of viable seed out of garlic mustard, which is what you care about when you're controlling garlic mustard. You want to stop that seed production. It does not eliminate the seed production, but it can be used as a method, and you can continue to do this even after it starts flowering. You can still have success, and it should be kind of one of those tools that you use in your toolbox when you're controlling these. And so now we've kind of adjusted our recommendations a little bit for garlic mustard. We still like fall treatments to the rosettes. Torching them with propane torches in the fall works well. Spring foliar treatments can go all the way up through early fruit development, we're finding. And then hand pull is effective still. Even you can do hand pull if you want, up until those fruits start drying, and at that point you don't want to mess with them because you're just spreading the seeds around. But again, I think it opens our window up and gives us 2 or 3 or 4 more weeks of time that we have more options to control in the spring. Alrighty, the second one I wanted to talk about today is Japanese stiltgrass. So Japanese stiltgrass is a major problem. I'm sure that if you've hiked around Southern Illinois, you've seen this plant. It's really becoming one of our worst problems here in Southern Illinois. It's an annual, and that's important for this research. Warm season grass spreads aggressively through bottomlands, riparian areas, disturbed woods, a very, very high priority for all of Illinois, for sure. See the map here, it's well established in the southern part of the state, but it's found all the way throughout Illinois, scattered, and even into some of the more northern states in the Midwest. So it's becoming more of a problem even outside of Southern Illinois. But we're concerned about it because of just its ability to form these dense stands and really crowd everything out, change the soil chemistry. There's a lot of negative impacts from this plant. And so when you're managing an annual plant, very similar to the biennial plant with garlic mustard, your goal is to stop seed production. That plant's going to die at the end of the year, whether you control it or not. So you don't really care necessarily about killing the plant. You care about stopping that seed production. And you can do that with a bunch of different ways: mowing it at the right time, pulling it, burning it, weed whipping. As long as your timing's correct, you can really reduce or eliminate the seed. And then you can use multiple types of chemicals for herbicides: contact herbicides, which just burn down the leaf tissue that it touches; systemic herbicides, which will translocate to the roots; or pre-emergent herbicides. And pre-emergent herbicides are a particular group of herbicides that really only work on seeds as they're starting to germinate and starting to get that radicle growing out. It'll kind of stop that. So they're very, very targeted. And so that's what I'm going to talk about today, is this idea of using a pre-emergent herbicide to stop the seeds from germinating in the spring. And so you can see just stiltgrass, again, this is kind of later in the year, full of seed heads. It'll die back. It'll look like this in the winter. And then early spring and March around in southern Illinois, you're going to start to see a lot of germination coming up there, and all of these little seeds really germinating and starting next year's that year's infestations. So our goal was to look at how effective are these pre-emergent herbicides for treating Japanese stiltgrass. Can they be used? What are different rates and different herbicides? What's the most effective? And then in particular, we're looking at how can you control stiltgrass while minimizing some of the non-target impacts of using this kind of herbicide. And so we did this here in partnership with Bayer, and we had 2 by 5 meter plots of a heavy stiltgrass infestation. We applied the herbicide just before germination in the spring, and then we watched it throughout the year. And we kind of collected visual estimations of the percent cover for stiltgrass and native vegetation at 1, 2, 4, and 6 months after treatment. And we used a bunch of different herbicides, a bunch of different pre-emergent herbicides, to try to figure out which ones may be the most effective. And we looked at a bunch of different rates. So we did some tank mixes, we did a bunch of different rates, as well as an untreated control. So we're really kind of just exploring and seeing what are what are the options out there. This is the site where we did this, and you can see has some native plants in there, but it's pretty heavily infested with stiltgrass. It's a pretty bad infestation. It's actually an old ash plantation here on site. So this is us applying. We marked out our plots in the spring, and then we applied the pre-emergent herbicide early in the year, and again to stop the germination of these. And this is what they looked like, a few months into it. You can really see our plots here, and then the stiltgrass doing fine in between our plots. And then just one more look here. We have one plot out by itself and amongst a sea of stiltgrass. And so we had a lot of data from this. This is the data that shows all the different treatments and the percent cover throughout the year. And so what you can notice right away is several of these treatments were very effective early, but then their effectiveness they lost effectiveness as time went on in the year. And that's important for stiltgrass because it has a very long germination window. It doesn't just all germinate in the spring and then stop. It can keep kind of germinating a little bit throughout the year and almost wait out and outlast some of these herbicides. And so the Esplanade had a couple different rates. Again, looked really good early, but then the stiltgrass recovered, and the prowl was the same thing. You can see the untreated there on the left. Stiltgrass quickly rose to 80% cover and stayed between 80 and 92% cover pretty much throughout the entire growing season. So all of these reduced the stiltgrass some more than others. And you can see down here between the Esplanade Sure and all the way in the Oust, all of those looked fairly effective at controlling stiltgrass. It kept stiltgrass below 10% throughout the year. But this is, again, only half the story. So this is controlling the stiltgrass. We also wanted to see what happened to some of the native plants. So if you add that data onto this chart, and so here is 6 months after treatment, percent cover of stiltgrass and percent cover of native vegetation. And you can see some of these, like the Oust at 2%, or the tank mixes, anything with Oust in it, did a great job at controlling the stiltgrass. It's the orange bars there. But it also really kept the native vegetation down. And basically those plots were bare the entire season. And so while it was good at controlling stiltgrass, we thought it was the level that it controlled all vegetation and knocked back the natives was worrisome to us. So we actually found that this lighter rate of Esplanade Sure, so 4.5 ounces per acre, did the best job, where it controlled the stiltgrass, but it let the native plants kind of come back the best. Another thing we noticed was anything that had the Oust herbicide in it was a bit of a concern to us, because what we found was it tended to move a little bit from where we put it down. So here is this shot of this plot again, and you can see this trail and this trail here marked in red. That's where rain flow kind of happened sometime during the growing season and actually moved the herbicide a little bit off. And so this is kind of moving downhill. So we have these streaks of bare ground kind of coming out of that, and that's overland flow moving the herbicide. And that's not what we really wanted in a natural area. And so we decided that Oust in this application was probably not our best herbicide, because of even though it does a great job of controlling stiltgrass, we didn't like that potential for non-target impacts in that movement. So what, kind of in summary, we found that the Esplanade 200 SC and the Prow were ineffective. They didn't control them. And then anything that had Oust XP was very effective, but we had these non-target impacts, so we didn't like it. So that the Esplanade Sure, of all the different herbicides we tested, really gave us the best control, and particularly at that 4.5 ounces per acre rate, where it controlled the stiltgrass and minimized these impacts to the native vegetation. And so the control recommendations now for stiltgrass, we've changed them around a little bit. Before germination, Esplanade Sure at 4.5 ounces per acre is a great pre-emergent to use. If you don't want to treat it at that point, late summer before it flowers, you can do low mowing or weed whipping, hand pulling small infestations, starting to look at growing season burns as an option, or you can control it with something like glyphosate or a grass-specific herbicide. So there's a lot of options for you to do stiltgrass later in the season as it gets close to flowering. But early in the season, this pre-emergent can be a tool that you can use. And the last of the species I'm going to talk about is Amur honeysuckle, or bush honeysuckle. It's one of the bush honeysuckle species. I consider this the highest priority invasive plant we have in Illinois forest in terms of potential for damage and change to the ecosystems. And that's largely due because it's highly shade tolerant, and can invade high quality, fairly intact forest. And I'm sure you're familiar with this plant. It has the delayed phenology, where it'll stay green much later in the fall. It'll green up earlier in the spring as well. It has the bright red berries that are bird dispersed. It's fairly ubiquitous, unfortunately. It's widespread throughout much of southern Illinois. I think we have some of the old growth first plantings of this in southern Illinois here at the Ag Station. We have some huge honeysuckle infestations that are just really big plants. And I found this one earlier this year that would qualify for a record, I think, as the biggest honeysuckle I've ever seen. It's quite impressive. But this plant, there's some conventional recommendations. People talk about using a cut stump or a foliar application, Glyphosate is really proven to be generally very effective at this one, but you see triclopyr, so that's Garlon, or there's a lot of generics. You see it recommended a lot, but then talking to a lot of land managers and then just seeing it ourselves, we often get very mixed results with triclopyr. So we wanted to really look at, is it an effective herbicide for honeysuckle, or should it be removed as a recommended herbicide is because it's not that effective? So what we did with this one was we wanted to test the effectiveness of triclopyr as a foliar applied herbicide to honeysuckle, to Amur honeysuckle. And then we also looked at some other herbicide options to see, are they effective or not? And so we wanted to look at this one in a longer scale. A lot of times some research just looks at how quick it kills it or how quick it defoliates, or maybe just 1 year. So we actually followed these and wanted to look at them for 2 years after the treatment, because there's some observational stuff out there that sometimes they look like the herbicides work really well, but in that second year, the plants will actually start recovering. So that's why we went 2 years to watch these plants. We did individual plant treatments. So we did 9 replicates for each treatment type. And this was our research here, but this was actually replicated in Indiana and in Wisconsin with some of my colleagues. So it's spread out. I'm only going to present our data here, but just know that it was done in multiple sites. And we wanted to look at a late summer fall treatment. We did ours in 2017, and then we followed up and measured percent defoliation at 1 year after in 2018 and 2 years after in 2019. And then we calculated mortality. So we decided if after 2 years it was 100% defoliated, there was no green leaves, we considered it at that point a dead plant. So we did triclopyr, we did Method, which is one of the newer herbicides, at 3 different rates. We looked at Milestone and then an untreated control for this. And so what we found with this one was 1 year after treatment, the Garlon, the triclopyr, defoliated at about 50%, but that dropped, and the plants actually recovered by the second year, and there was only 30% defoliation. Milestone was not very good, and you can see the untreated control, they pretty much did fine. There was 10% or less defoliation the whole time. And then Method, we had to kind of get to that higher rate, the 1% Method, before we really started seeing control rates that approached what we normally see with glyphosate. And this is what it looked like. So this was a plant that was treated 2 years prior to this picture with triclopyr. And you can see there's parts of the plant that are dead, but a lot of that plant is still living. There's some sprouts coming from the bottom. And this was pretty common what we saw in terms of initial defoliation, and then it started recovering already in that first year. And by that second year, it was very, very well recovered. It kind of grew out of the damage, unfortunately. And then the lighter rates of Method, those also not effective. This is 2 years after treatment. That plant looks fine, almost no defoliation. But once you got into the 0.5% or 1%, those plants usually were dead 2 years after treatment. They usually did a pretty good job. This one's very dead. The 0.5%, this one, 1% was dead, and it's starting to get taken over by stiltgrass and chaff flower even. So it was very, very dead. So again, we're worried about mortality. We want to kill these plants. We want to kill the root system because this is a perennial plant. So after 2 years, we calculated mortality, and out of all the plants we treated with Garlon, 10% of them were actually died. So 90% of the plants that were treated with Garlon, that the triclopyr ended up living. The same goes for the Milestone. So we decided that, yeah, our observations were correct, that the triclopyr is just not a super effective herbicide to use for honeysuckle, even though it's recommended a lot. The Method at 1% was highly effective, 80-some% mortality. And again, this was the data that we had here. If you look at the data from the other sites across the Midwest, it mirrored this very closely. And so kind of the summary of that one, triclopyr treatment was ineffective. Even though it defoliated it right away, almost every plant sprouted and continues to grow. Milestone was also ineffective, but the method was highly effective at that 1% rate, and it can be another tool for our toolbox along with glyphosate or some of the other mechanical controls. So our recommendations for honeysuckle, for Amur honeysuckle, bush honeysuckle, is basically stop using herbicides that are triclopyr based for controlling honeysuckle. It just, it may look good right away, but it's not giving you the results that you want. Glyphosate still is the most effective herbicide, but this method at 1% is an alternative that is proven to be effective and safe. Also weed wrenches, mechanical control for smaller stems, prescribed fire and grazing may be options as well. They certainly knock it back. And then there's some new work out there. Aerial control in the fall may be an option, and there's some neat research coming out on that too. And so just in general, research is vital to extension. We try to get this information into the hands of people that need to use it. Our research here, invasive plant research, we're going out there trying to determine the most effective and safe means to control these invasive species. We're looking at herbicides. There's a lot of other methods as well. And so we're trying to do this with looking at a bunch of different invasive plant research projects. And that is just a really quick rundown of what we've got going on here at the Extension Forestry.